Okay, we'll start the sixth topic, which is antibodies, and we'll understand the different types of the antibodies in our body and how they are differentiated from each other. So let's talk about the definition. So antibodies actually are mucoproteins and found mainly in the gamma globulin fractions. So we'll understand later what are the different fractions of our serum. And they identified this and they named them according to their migration on the electrophoresis. So we have generally serum proteins, and clinicians can obtain information about the disease states or the patient's general status by measuring the concentration of these proteins. The normal serum protein level ranges from 6 to 8 grams per deciliter, and generally you have two fractions. Albumin, which occupies most of it, as you can see, these actually represented the electrophoretic pattern, and you can see that albumin occupies. Okay, for you to understand, the thicker and the heavier, the band will migrate slower, and hence will appear on top of the gel. So you can see that albumin occupies most of the concentration of the serum proteins. While the globulin, they named it depending on their migration pattern. So globulin actually is divided into 4 groups based on the electrophoresis. You have the alpha 1, you can see this is alpha 1, and actually these are the concentration of the alpha 1, 0.1 to 0.4, and you have the alpha 2, and you have the beta, and you have the gamma. So now you understand now why they named them alpha 1, 2, beta, gamma. So they named them according to the migration pattern of these globulin proteins. So we can see they can represented as this line chart, which shows the albumin, which is peaking, and this actually correspond the heavier band of the albumin. And you can see the different bands for alpha 1, alpha 2, beta, and the gamma. Okay, and this gamma actually, which represent all the different types and all the different classes of the protein of the antibodies. And they generally have given the name because originally when human antibodies are injected into animals, they become antigenic and the animal respond to these antibodies. And the result anti-human antibodies can be grouped into 5 different classes, which is IgG, which is the type of the antibody which is most abundant, and IgA, and IgM, IgE, and IgD. And we'll understand later on why they are named GAMED. So again, here you can see the electrophoretic pattern, and you can see the major line, which represented the total serum protein. And you can see actually this major line can be divided into sublines. And you can see that in the region of the gamma globulin, you can see the different antibodies just below this line. So it's kind of like an average of this line. And for convenience, you can always remember the concentration of different serum proteins by the way you show your fingers in that way. So you can see the how the albumin differs from alpha 1, alpha 2, beta, and gamma. So you can see here how is the difference between the normal pattern of the different serum protein, the serum protein electrophoretic pattern. You can see this is a normal pattern, and you can see, for example, how this pattern is changed in a case like multiple myeloma. So you can see there is an increasing, majorly the increase will be in the gamma region, and you can see as well increase in the alpha region, alpha 2 region, and alpha 1 region, and beta region. So you can understand now for certain diseases or for certain cases, you can identify if there is a condition by measuring the pattern of the serum protein. And here as well, you can see again, this is a normal pattern. You can see the albumin, and you can see the different fraction. And this is a gamma as well, showing the IgA, IgM, IgG. And of course, when you compare this for different cases, you can see how this pattern is dramatically changed with different condition of diseases. For example, you can see in the nephrotic syndrome, you can see how this pattern is changes. The globulin, the albumin fraction have been decreased, while the alpha-2 globulin have been increased. While in cases like delayed response pattern, you can see how these pattern have been changed. So let's see here the structure of antibody generally before going into details. So you have the antibody, generally it have two long chains and two light chains. Actually, the long chains is attached, the heavy chain actually is attached by disulfide bonds, and as well the light chains are attached by a single sulfide bond to the heavy chain. For each heavy chain or light chain, you can see that they have a constant region of the same that share actually why it's constant, because they are actually the same on in all the different antibodies, while it has as well a variable region which gives the specificity of each type of antibody. As well for the light chain, you can see actually it consists as well from constant and a variable region for the antigen binding site. So you can see that the combination between the variable region of each heavy chain and the light chain share together an antigen binding site for recognition of the antigen. And we'll talk in details later on the structure. You can see the different types of antibody. You have IgG, IgE, IgD, IgA, and IgM. You can see as well that the antibody can be located on the surface of the beta cells, and you can understand that these cells actually in this state, it is not still a terminally differentiated cells, which is the plasma cells. And this antibody on the surface of B cells can bind on antigen, even if it's the antigen on a microbe. If you look closely into the structure of the antibody, actually, as we said, it consists of 4-chain protein with 2 heavy chain H and 2 light chains L chain polypeptides. And they are linked, as we said, by disulfide bonds. If we talk about the heavy chains, you can see as well, actually the constant region is divided into 3 constant regions, abbreviated with CH, constant heavy. So you have 3H constant regions, CH1, as you can see from the figure, CH1, CH2, CH3. For the light as well, they have 1 constant region, CL, constant light, and you can see the variable region of the heavy change. Actually, it is variable because actually it is can be variable or varied from 5 classes, which is called generally isotypes: the gamma, the alpha, the epsilon, the delta, the mu. So actually, if this variable region, that's why it came the names. So gamma gives the IgG, epsilon gives the IgE, and so on. For the variable light regions, you have 2 isotopes, either kappa or delta, I guess. So for the C domain, for the domains, you can see that the variable heavy region, together with the variable light region, both of them, they are actually, we can call it the hypervariable region. And these 2 combined region can form the epitope binding site from the antigen. So the 2 epitope binding site region, the 2 epitope binding region are known as the antibody idiotype. So when you collect all these 2 of them, it will be called the idiotype. And there is a hinge region which provide flexibility between this region of the antibody and the FC region. You can see as well the location where the carbohydrate can bind to the antibody. So let's go closer into the structure and see. So we have the antibody originally is formed actually from FAB region, Fab region, which is actually the variable region, and FC region connected together with the hinge, as we said. So if we enlarge this part, you can see some terminology that you need to be familiar with. So you have the idiotype. Actually, it's the one part of the epitope binding site, and you have the idiotopes, and you have the paratopes, which actually shares its part in the epitope identification, joined by the two variable region from the light chain and the heavy chain that can recognize the antigen. So basically, you have three expressions now: the idiotopes, which you form the idiotype, and together the idiotype with the other idiotype will form the epitope binding site with the help of the paratope. Okay. So here again, you can see the different structure of the antibody. You can see that the fragment antigen binding site, you can see all this region is the fragment antigen binding site. Then you have the fragment crystallizable, or actually people say constant region of the antibody. You can see where is the constant region of the heavy chain, and where is the constant region of the light chain. You can see the hinge as well. You can see where is the variable region of the heavy chain, and you can see where is the variable region of the light chain. As well, you can see the site of the binding of the carbohydrate chain. You can see how they differentiate between the hypervariable region and the variable region and the constant region for different type of chain. Actually, this figure is another illustration of the structure of the general structure of the antibody. So here again, you can see the difference between IgG and IgA. So you can see the percentage of, you can see there are subclasses actually of IgG. You have IgG1, IgG2, IgG3, and you can see the different percentage of each of the IgG. You can see as well the percentage of this type of the antibody in the serum, as well as the half-life time. You can see that there are each type of antibody. You can see that IgA can do a dimerization by binding through a J chain. And you can see as well the IgM, it can exist as a pentamer, consists of 5 similar molecules. And this one can be bound to the mature B lymphocyte. So understand now that the B lymphocyte can bound to the IgM. And you can see as well the concentration and the half-life time of this type of antibody. For IgGD, as well have the capacity to bind to the mature B lymphocyte, although the percentage of it and the half-life is much lower than the IgM. For the last type, you can see that it is a totally different antibody, IgE, and actually related to the basophil granulocyte, and they have very low amount of concentration in the serum, and it is the least time as a half-life. So actually, each class of this antibody totally have a different function, although all of them are related. So let's talk about special types of antibody, actually mostly generated in the lab, which is called the monoclonal antibody. So what are the monoclonal antibody? These actually are antibodies of a single specificity. They are highly usable and can be used and desired for many purposes. They can be included in a specific diagnostic test and immunotherapy. So actually it can be made, these type of antibodies can be made routinely by fusing the splenic B cells, the B cell isolated from the spleen, from an immunized animal with malignant plasma cell, forming some sort of called hybridoma. So you take the advantage of a malignant plasma cell, which you have a continuous advantage of secreting antibodies, and you fuse it with the splenic B cells which will generate the different types of the B's, as you know, B cells differentiated into plasma cells, which secrete antibody. The hybridoma generated is very efficient in continuous secreting of an antibody of a specific epitope. So actually how it works actually, so you can see the B cell hybridoma secrete the desired antibody, what you actually want, can be isolated from the other antibody by reactivity with the antigen of concern. So once the hybridoma starts generating antibodies, it will have you will have different antibodies. Then you can actually separate your desired antibody upon exposure to a specific antigen. Then this antibody can be cloned, and by the hybridoma can be cloned and expanded in the tissue culture, resulting in a very large amount of antibody of a specific single specificity. So we can see in the figure, so we can see here is a mouse. You inject the mouse with the antigen, and this mouse internally, it will produce a plasma cell that carry on its surface the antibody specific to this antigen. So then you bring a tumor cell, which actually will be a malignant tumor cell, and you fuse together with the plasma cell. You will generate a hybridoma. So you will gain the advantage of having the plasma cell, which have a continuous secreting of antibody, and the advantage of the tumor cell, which have a continuous cell division. So you will have a fused cell that have the capacity of continuous dividing and continuous production of the desired antibody for that specific antigen. And this will give you a monoclonal antibody because it's only specific to the one epitope. So you can see here, let's talk about the immunotoxins, how these are related to the monoclonal antibody. So the monoclonal antibody can be constructed using this antibody with the domain which is specific for the cancer antigen. So in this scenario, the Fc domain is removed and displaced by the toxin of choice. So such antibody can bind specifically to the cancer cell antigen via the mouse Fab hypervariable region. So it's a kind of recruiting the lethal toxin or the drug to the target cell or the cancer cell. So in this figure, you can see the cancer cells can express a lot of specific markers or specific protein to this antibody. So you can target the antibody and you can lock this antibody, can identify this specific receptor on the cell of the cancer cell. So in this scenario, you can link the lethal drug for this cancer cell so that the antibody will recruit this drug to the cancer cells. And in this scenario, you will spare the normal cells. You will only specifically attack the cancer cell. So you can see how this occurs. So actually you have the antibody, this actually the basic structure of the antibody. So you can have the cytotoxic agent that will kill the specific cell of interest or the target cells can be linked to the antibody, to the monoclonal antibody via a linker that attach to the cytotoxic agent to the antibody. And this linker should be designed to be stable in the circulation and is not affected. And once it reach the target cell, it can release this cytotoxic agent to kill the target cell. So let's see here another level of the use of the monoclonal antibody, which is called the bispecific antibody. So this actually increases the efficiency of immunotoxins. And actually, this kind of antibody is engineered from two different monoclonal antibodies. So you can see here, this antibody, it looks different because it came from two sources. They are engineered to identify two different epitopes. So why they engineered this kind of antibody, and still it's called a bispecific antibody, to enhance the efficiency of the immunotoxins. So you can see how this works. So initially, this antibody will have an epitope antigen recognition site that will recognize CD3, which usually is displayed over the T cell. And it will have the other epitope identification site for identification of the tumor antigen on a tumor cells. So for first time, it will provide the strategy of recruitment of the tumor cell to be very near of the T cells, make advantage of this close relationship in the process of destruction or the activation of the immune system toward the tumor cells. And usually this antibody, this bispecific antibody, is engineered so that the Fc region will bind to the Fc receptor on an accessory cell that help this system, which might be macrophage, dendritic cells, or even NK cells. In this scenario, and when all these binding occur, this will activate the process of binding between the dendritic cell or the antigen-presenting cell with the T cell, will lead to physiological stimulation of T cell to be activated. And hence, once it is activated, it will it will secrete factors that lead to the lysis, especially if it's a cytotoxic T lymphocyte. It will release the factor that lead to the lysis of the tumor cells, as well you can see that so as well, the antigen-presenting cells, it can mediate phagocytosis of the tumor cells through the process of ADC. So to summarize this figure, the use of a bispecific antibody, it will enable the interaction between the T cell and the tumor cell and the antigen-presenting cells. The activation of the T cell will lead to the secretion of lysis factor that these cytotoxic cytokines will lead to the lysis of the tumor cells. And as well, this scenario will provide will enable the dendritic cells or accessory cells to do the antibody dependent cytotoxicity toward by the process of phagocytosis. It's a process of opsonization, if you remember, so that they can do phagocytosis of the tumor cell. So let's see here a video to describe more details about the monoclonal antibody.
When it comes to treating diseases like cancer, modern medicine has an impressive arsenal. And one of its most versatile weapons are Y-shaped proteins called monoclonal antibodies. Our immune systems already produce their own antibodies. They come in billions of variations, each matching a specific target, such as a particular toxin, bacteria, or virus. When they bind to their target, they send a signal. This bacterium is now marked for destruction. These naturally produced antibodies are pretty effective, but in the 1970s, scientists figured out how to mass produce them. They can take any specifically targeted antibody and make huge numbers of identical clones, which means you can produce monoclonal antibodies for almost any disease you can think of. Which brings us on to cancer. Mutated cells that start growing out of control can be deadly. But the body's immune system isn't defenseless. Meet the CTL. It's a type of white blood cell that can detect infected or mutated cells. And when it finds one, it secretes toxic molecules that initiate a self-destruct sequence. But the CTLs do sometimes need to be reined in. If they get over-enthusiastic, the immune system can start causing collateral damage, attacking healthy cells. Fortunately, the CTLs can be turned off by activating a molecule called PD-1.
So here it's very important to understand that the CTL must be regulated. So this PD-1 molecule, you can see, it is switched on to avoid the hyperactivity of CTL, because they found that the hyperactivity of CTL can lead to a sort of autoimmune disease, and the immune cells, the white blood cells, start to attack our own cells. So this CTL must be regulated through what's appear in the animation here, which appear like the PD-1 switch should be switched on. And here we can see, later we'll see in this video, how the cancer cell can manipulate this switching on or regulation capacity of CDL for its advantage. So by playing around with the BDN1 switch or receptor. Let's see.
Trouble with having an off switch is that cancer cells can exploit it. Many cancer cells protect themselves with a counter receptor called PD-L1. This is where monoclonal antibodies come in.
So you have seen now that cancer cells, it make advantage of this PD-1 receptor, and actually they done switching off for CTL. So in case this cancer cell is switched the CTL, so now CTL will not identify any cancer cells. And here comes the technology of the monoclonal antibodies trying to release the capacity of the CTL through activating again the CTL toward the cancer cell. So as we will see now.
These come in. Scientists can use one of two types. One that binds the PD-1 molecule, or another that goes for PD-L1.
So here, scientists have managed to generate two different monoclonal antibodies. So you can see one of the antibodies will switch on back the PD-1 receptor protein on the surface of CDL, and the other monoclonal antibody will bind to the PD-L1, actually represented here from the arm. It's another receptor protein on the cancer cell that is used to switch off the CTL. So these two types of antibodies will efficiently activate the CTL.
Thanks to the monoclonal antibodies, all of the CTLs are fully activated and can blast away the cancer. This is just one way that monoclonal antibodies can be used. They've already proven effective against a whole range of cancers, and yet more are still being developed. With their ability to be targeted at specific diseases, monoclonal antibodies are turning out to be a game changer, and one of our most effective weapons in the fight against cancer.
Okay.
Antibodies are the warriors inside our body. They are part of our immune system, recognizing and fighting against bad foreign invaders called antigens. Antibodies can bind to a broad range of antigens and are produced by cells of the immune system, known as B cells. To learn more about antibodies and antigens, watch this video. Antibodies can be used for medical and diagnostic applications in the form of monoclonal antibodies. So what are monoclonal antibodies? The word monoclonal means a single clone. Monoclonal antibodies are identical antibodies produced in laboratories from a single clone of immune cells. The cloned immune cells are genetically completely identical, and so the antibodies they produce are also identical to one another, which is perfect. To work properly, we need large numbers of identical antibodies. Monoclonal antibodies have a high specificity. which means they can recognize and bind to a single antigen binding site. This means they are targeted to a specific antigen and is a key trait that makes them useful in diagnosing and treating diseases. So how are they produced? Well, first off, we need an immune system, which is why we use mice. In a laboratory, a mouse is vaccinated with the target antigen. This stimulates the B cells found in the spleen to produce antibodies against the target antigen. The spleen of the mouse is then removed. The B cells in the spleen are isolated and fuse with a tumour cell. We need to fuse the antibody-producing B cells with a tumour cell because the B cells don't have the ability to divide, but tumours, however, do divide easily. This fusion cell is called a hybridoma cell. The hybridoma cells reproduce rapidly to make cloned cells, which all make the same antibody. These monoclonal antibodies are collected and purified, ready for use. They match up perfectly with the target antigens in the body and are made in huge quantities. Monoclonal antibodies are potentially really exciting and can be used in a number of ways. I bet you didn't know antibodies were used in pregnancy tests. Monoclonal antibodies on the pregnancy test stick bind to the HCG hormone found in the urine of pregnant women, causing a colour change. Monoclonal antibodies can target the antigens on cancer cells and be used in a number of ways. Firstly, by combining them with an anti-cancer drug, which can then accurately locate and target only the cancer cells or avoiding the healthy cells. Secondly, for detection, they can carry special markers which show doctors where the cancerous cells are starting to build up. Thirdly, they can be used to trigger the body's own immune system to recognize and kill the cancer cells. They can also be used to identify other diseases and locate blood clots. So those are a few uses, but are they all good? We've seen the positives, so what are some criticisms? They are expensive and time-consuming to produce. They have more side effects than expected, and they are too specific, meaning that whilst yes, they can diagnose and attack diseases, they only can attack one type each time. A more diverse and adaptable solution would be better. The potential power of monoclonal antibodies is clear. They are not yet as widely used as everyone hoped when they were first developed, but scientists are working hard to make them better and safer for wider use. Their journey is just beginning.
If you liked the video, give it a thumbs